Hello and welcome. Now you will discuss some key bioinformatics concepts: base calling, error probability, and quality score. Previously, we discussed some other key bioinformatics concepts in detail. Now let's connect that knowledge to an important step called base calling. Base calling in Illumina NGS is the step where raw optical signals from the sequencer are converted into actual DNA bases A, T, C, and G. As we discussed earlier, this happens during sequencing by synthesis and involves signal detection, image processing, and probabilistic interpretation. And finally, output generation in the form of FASTQ file. Now, let's see how this works step by step. In Illumina SBS, after each sequencing cycle, the instrument captures images as we can see here. Then the software identifies the location of each cluster and the fluorescence intensity at each cycle as we can see here. This step is known as cluster detection and registration. Next is intensity measurement. For each cluster, the fluorescence intensity is measured in all four channels A, T, C, and G. For example, A might have the intensity of 1200, T might have 150, C might have 90, G might have 110. We can see the summary of signal intensity in this table. Now comes the key step, base calling. The system determines the base based on signal intensity. The base with the highest intensity is initially selected. So in this example, since A has the highest signal, the called base is A as we can see here. But in reality, it is not that simple. Advanced algorithm also consider signal overlap between channels, background noise, and phasing and pre-phasing effects. So base calling is not just picking the highest signal, it is a statistically informed decision. Now let's understand phasing and pre-phasing that we have mentioned few moments back. Over sequencing cycles, not all DNA strands behave perfectly. Some strands fall behind, this is called phasing. Some strands jump ahead, this is called pre-phasing. Here, for simplification and explanation purposes, the DNA extension is shown upward. However, in sequencing by synthesis or SBS, DNA extension actually occurs towards the flow cell surface as we have discussed and shown earlier. First, look at the image on the left side. As we can see here, all strands synthesize exactly 4 bases by cycle 4, reflecting perfect synchronization, and the signal intensities are strong and clean for all cycles as we can see here. Here is the addition of 4 bases. Now, not all strands behave perfectly as we have said earlier. If we see here, some strands don't complete a base addition. As a result, by cycle 4, they are at base 3 as we can see here, here and here. The main signal is contaminated by this lagging signal. This is called phasing. As a result, the signal intensity at cycle 4 becomes weaker and mixed due to the contribution from out of synchronization strands as we can see here. On the other hand, few strands incorporate the next base earlier than expected as we can see here. So by cycle 3, some strands appear to be at cycle 4. The main signal is contaminated by this ahead of cycle or skipping signal. This is called pre-phasing. As we can see here as well as here, the signal intensity at cycle 3 becomes less distinct due to mixed signals from the strands at different synthesis stages. What does this cause? It leads to mixed signals because different strands in the same cluster are now incorporating bases at slightly different times. To correct this, the system uses correction algorithms. These algorithms deconvolute overlapping signals and separate the true signal from noise. As a result, they improve the accuracy of base calls, especially in later cycles. Now let's move to the next important step, quality scoring. After a base is called, the system does not stop there. Every base call is a prediction based on signal, and every prediction has a small chance of being wrong. Each base call is assigned a quality score, also known as a Fred quality score or Q score. So what does this quality score represent? It represents the confidence in base call. More specifically, it is based on the probability that the base call is incorrect. The scoring system uses Fred quality score. The Fred quality score is logarithmically related to the probability of base calling error or P error. We will discuss base calling error probability later in this lecture in detail. The relationship is given by this equation here, as we can see. Now, what does this mean? If the probability of error is low, the quality score is high. If the probability of error is high, the quality score is low. Let's look at some typical interpretations. For example, if Fred assigns a Q score of 30 to a base, this is equivalent to the probability of an incorrect base call 1 in 1000 times. As we can see here, this is the Fred quality score of 30. This is the probability of incorrect base call. This means the base call accuracy, that is probability of a correct base call, is 99.9%. When average sequencing quality reaches Q30, most bases are highly accurate, that is 99.9%, and virtually all of the bases will be near perfect, having almost zero errors and ambiguities. This is why Q30 is considered as a benchmark for quality in Illumina next generation sequencing. Similarly, a Q score of 20 means about 1 error in 100 or 99% accuracy. So higher Q score means higher confidence. Now let's understand probability of error. What does it mean? After a base is called A, T, C or G, the system estimates what is the probability that this base call is wrong. This is called probability of error or P error. This probability is not directly calculated from raw intensity using a simple formula. Instead, it is derived from signal quality and model-based prediction. So now, how does Illumina estimate this probability? It uses statistical models trained on real sequencing data. So rather than a single equation, multiple factors are analyzed together during sequencing in modern Illumina systems. First factor, signal intensity comparison. The system compares the intensity of the chosen base with the intensities of other bases. If one channel is much stronger, then the confidence is high and probability of error is low. But if the signals are close, then the system becomes uncertain and the probability of error increases. Let's look at this example. If A has the intensity of 1200 and others are very low, as we can see in this table, 50, 40, 30, then the confidence is high. Now look here, if A is 400, T is 350, C is 300, G is 280, then the confidence is low as the signals are very close. Next factor, signal to noise ratio. If the signal is strong and the background noise is low, then the base call is reliable and probability of error is low, as we can see here. But if signal is weak or noisy, then the probability of error increases. Another key factor, phasing and pre-phasing. During sequencing by synthesis, some DNA strands fall behind. This is called phasing, as we have said earlier. As we can see also here, some strands jump ahead. This is called pre-phasing, as we have mentioned earlier also. So what does this cause? It leads to mixed signals across cycles. So the signal you observe is actually a combination of multiple states. To handle this, algorithms estimate how much signal contamination exists and adjust the confidence accordingly. Next factor, crosstalk between channels. Fluorescent dyes are not perfectly distinct. Sometimes a signal from one dye can bleed into another channel. Correction models try to fix this. They adjust intensity value, as we can see here, with crosstalk and after crosstalk correction. But some ambiguities still remain, which increases the probability of error. Finally, machine learning and empirical models. Modern base callers such as Illumina's real-time analysis software use large training dataset. They model error probability based on intensity patterns, cycle number, cluster behavior, as we can see here as well as here. So in summary, probability of error is estimated using signal comparison, noise levels, phasing effects, channel correction and statistical modeling. If the signal is clear and well separated, the probability of error is low. If the signal is mixed or overlapping or noisy, probability of error is high. Now let's look at the key factors that affect error probability. There are several important factors that influence how confident a base call is. First, signal quality. If the fluorescence signal is strong and clean, then the base call is highly reliable. but if the signal is weak, the probability of error increases. Second, cluster density. If the clusters are too close to each other, their signals can overlap. This makes it difficult for the system to correctly distinguish between them, and this can lead to miscalling of bases. Third, cycle number, also known as read length effect. In early cycles, accuracy is usually high, but as sequencing progresses, signal intensity decreases and phasing effects accumulate. So in later cycles, the probability of error increases. Fourth, phasing and pre-phasing that we have discussed several times. During sequencing by synthesis, some strands fall behind and some move ahead. This effect accumulates over cycles and it becomes a major source of error, especially in long reads. Fifth, sequence context. Certain types of sequences are more difficult to sequence accurately. For example, homopolymers, G-series regions, and regions forming secondary structures. This can reduce signal clarity and increase the probability of error. Sixth, optical noise and instrument factors. This includes camera sensitivity, background fluorescence, and laser fluctuations. All of these can introduce noise into the signal detection process. 7. Reagent quality. Sequencing depends heavily on chemical reactions. If there is poor incorporation efficiency or incomplete removal of fluorescent levels, then the signal accuracy is affected and error probability increases. The degraded reagents can also cause reduced chemistry performance and signal quality. All of these influence how confident the system is. So in simple terms, if the signal is clear and strong, probability of error is low. If the signal is mixed or noisy, probability of error is high. Any factor that reduces signal clarity or increases signal ambiguity will increase the probability of error. Now let's move to the final step, output files. After base calling and quality scoring, the data needs to be stored. First, data is stored in BCL files. These are raw base call files generated for each cycle of sequencing. Then these BCL files are converted into FASTQ files as we have discussed previously. FASTQ is the most commonly used format for downstream bioinformatics analysis. What does a FASTQ file contain? It contains the DNA sequence and the corresponding quality scores. So in summary, base calling converts fluorescence signals into DNA bases. It uses intensity measurement, signal correction, and statistical modeling to ensure that each base call is as accurate as possible. Each base is assigned a quality score based on probability of error. And finally, all base calls and quality information are stored in FASTQ files for further analysis. So now we have learned base calling. This is how fluorescence signals are converted into DNA bases A, T, C, and G. Probability of error. This tells us how likely it is that a base call is incorrect. Quality scores. These scores show how confident we are in each base call. Output files. Finally, BCL's files are converted into FASTQ files for downstream bioinformatics analysis. That's all for now. Thank you everyone.